Yes. Mm, Good morning. My name is Jeff Ochen. I'm presenting the feedback for the first session and the second session that took place on 29th January and the 1st of February. I take this opportunity to welcome everyone for this presentation. Mm, I'm going to make it brief. It is all about the bioinformatics of infectious disease that we learned previously for the two sessions that took place with the support of our mentor, as you see them in the screen. Yeah, we have four mentors that have been working hand in hand with us to make and understand more in bioinformatics in infectious disease. After that, I will start by giving the proposed purpose of the program. Sorry. Yeah, the purpose of the program is to learn how bioinformatics are used in the context of the infectious disease. After the end of the program, I hope I will be able to know how the bioinformatics are used in the context of the infectious disease. Uh, mm, the second one is the generation application of bioinformatics and other applications that cause to disease. And all the general applications of the bioinformatics and the other organism on how they cause the disease. I think by the end of this session, I'll be able to know. Mm, the third one is to learn how to get data, how to analyze data, and what we will do with our researched analyzed data. The data that we are going to get, how are we going to analyze it? Which we have various methods of data analysis. How are we going to analyze that data? And after the analysis, how are we going to use this data in the research, in the publications? Also, at the end of this program, I will be able to support those who are in leveraging bioinformatics tools through publication, also to enhance our career in research. We publish in public domain for the people to attain the knowledge on what someone's work. Mm, Thanks so much again. Also, I want to start by introducing bioinformatics, whereby we learn on the infectious disease. In infectious disease, it is diseases that spread directly from one person to another person. It's caused by pathogenic microorganisms. The pathogenic microorganism is as follows. We have the bacteria. These are the life organisms. We have the viruses. We have the parasites, and the last one is the fungi. Mm. Again, we were able to differentiate the infectious disease and the non-infectious disease, whereby the non-infectious disease, they are not caused by the pathogen, but can be spread, they can be shared through one person to another person. And they are caused by the environment, the environment that we live, the environment that the pathogens live, also the nutritional deficiencies, the type of food, our diet also cause this non-infectious disease. Also, the genetic inheritance. It can be from one person to another person through genes, whereby from the mother to the son, or from the son from the father to the son. Also, the lifestyle that someone chose also can contribute for non-infectious disease. We hand ahead and look on how the pathogen spread, whereby there was a replication. They spread through replication. They replicate, the virus replicates to multiply. Also in dead cell death. Yeah, the dead cells, whereby the pathogen spread through in dead cells. The fourth one is the third one is the organ function damage. Yeah, the organisms that are damaged, also they allow the pathogen develops and easily spread in the organs that has some damage. the relax of the toxin. That's the last one where the pathogen spread. Those four methods are the ones that we learn on which the pathogen can spread in the organisms. Again, we were able to understand what the omic logic is in bioinformatics, whereby the omic logic bioinformatics training programs are designed to introduce and help masters the application of bioinformatics to infectious disease research and study on how host pathogen interaction, the interaction between the pathogens. We will be able to explore relevant tools whereby the tools that we are going to use, the tools that we are going to learn, the tools that the interaction between the other people. They explore the relevant tools, method, and data repositories. Also learn from curative case studies and peer review publication. The previous studies that was done previously, we were able to interact with the previous studies, the previous data that had been published before. Also the peer reviews publications. The third one is to test your understanding by analyzing sample data. The understanding how you understand on the simple data that you are going to get, which methods are you going to analyze it. Also the master's key element of analysis and visualization tools. How are you going to make your data more understandable for the reviewers? You must present in a data in a manner that it is able to be understood and also to give a directives. We went ahead to know the various methods in the sequentials and alignments, whereby we have a sequential, variation, and alignments. In sequential, we tackle on how to perform multiple sequences analysis in important and identify importance variation in genomic losses study, variation between groups of genomes, whereby we were able to know the variation between the groups of genomes. The second one is the evolutionary and phylogenetic analysis, whereby they explore of evolutionary relationship between the virus, between the bacteria and the fungi genomics by analyzing alignment and phylogenetic tree construction, whereby the phylogenetic tree divides into two. The third one is the functionality and protein structure. The functionality and the protein structure. To explore the functionality and protein structure is to explore the physics, chemical, and structure aspect of virus, protein by mapping your analysis on 3D structure and finding stage interaction and similarities. The last one is transcriptomic analysis of host response. We have to develop a sense of host pathogen interaction, drug testings, and immune activation by analyzing the transcriptomic data into public domains. We also tackle on the picture that you are seeing here. The picture is it shows how the pathogen interacts and replicates through the genomic sequence. This picture I use the application for this process is a genomic sequence and the application of genomic sequencing. I hope you are seeing this is the interaction of the pathogen and then it replicates. It is visible. You can able to see. And the process is genomic sequencing, whereby the genomic sequences is we are going to use it to transform data sequencings into data files. We are able to get data through transformation into the data files. This one will also help us to study the problem expressions and also we were able to have a diagnosis whereby we will be use the DNA extraction through the polymerase chain reactions, which will help us for the diagnosis of the right drugs. Also, this one will help us to use for selling specific sequencings for strain detections. Mhm. We were also able to learn on specific sequencing for strain detection and outbreak investigation, whereby we can use this to know the outbreak of one or two various diseases. Also, the population structure and evolutionary history. Mm. Another important is the the protein expressions. Mm. Also, this picture help us to learn a lot in the pathogen application, pathogen interaction, and also the pathogen replication and how it caused harm to the human being and their effects. Or through genomic sequencing, we will be able to use various methods. After this, we also get a chance to interact with the server. In this server, we were told to open an account, whereby each and every person, after opening an account to join it, we are going able to interact much on how we are going to get the sources of data from different studies, whereby the source of data can be generated from the HIV, the malaria, and also the Ebola. We will be able to get the sources of the data from different studies on Ebola, different studies of in HIV, whereby you will use your pipeline. Yeah, my pipeline. The pipeline is all about something that connects multiple steps into sequential scripts, whereby once you created a pipeline, it has been added to your scripts. Yeah, the moment you create a pipeline, it is automatically being added to your scripts. After creating a script, then it goes to it add the value of the pipeline. Yeah. I'm very sorry, I will not be able to show how this process we can generate data from it, different data, because I'm presenting my 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 presentation on PowerPoints, but through web browser I'll be able to show how all the process comes in. Yeah, I think those are the presentation that I'm able to present. I'm much thankful for the training, and I'm capable that by the end of the training, I'll be able to have a very good presentation, have to explore, and also I'll be able to come up with projects that will enhance my career. Thank you.